Alright, here I'm going to go over worksheet 1, kind of the instructions and give you a few hints. Um, Everything's in the videos for these, but might not be obvious. There are a lot of videos this week. You may not have gotten to some of them at the end, and maybe you don't realize some things. But as far as the number 1 here on the worksheet 1, pretty straightforward. Hopefully everybody knows that DNA is double-stranded and has a complement. And so what I'm asking, it's pretty simple. I think everybody will know it. Essentially, I want to find the other strand. That's called the complement. And so that means, of course, if there's a C here, there's going to be a G down here. Let's see. That's the first one. Of course, I can't draw that small when making a video like that, but especially with a mouse. So, of course, this C is going to correspond to a G, the T to an A, the A to a T, and you just follow it across here for all 21 bases. Now, when you transcribe it to RNA, it's actually the same thing as up here. In other words, here we're still using A's. You can see that, C's, G's, and T's. Maybe I can use a text box with this thing. Well, next time. We'll keep going with this. But down here, of course, in number 2, we're going to replace the T here. Instead of using a T like we did here, you replace it with a U for uracil instead of thymine. In other words, we're going to transcribe it. Transcribe means, of course, you're not copying it. You are copying it, but you're making maybe a poor copy, if you want to think of it that way. If this were a Xerox machine, a photocopier, then you put it in, and if it makes really high quality copies, then you would call it a copy up here. That's finding the complement. But maybe it finds poorer copies, or one way to look at it, since this would end up, if you copy it, you end up making DNA, which would mean you're copying the DNA. Maybe it's like 2 strands would be like front and back of a page. So up here you're making a copy that's front and back, and it's color. That would be using the T. That we'll think of T as being color. But down here we're making only a single side copy, because RNA is going to be single-stranded, and instead of the result of, like up here, being double-stranded. And we could think of it as in black and white. And so it's a single strand, poorer copy in that it's black and white. You will think of as black and white. So you can think of it that way. So transcribe and complement, make sure you understand complement has to do with making a copy of the strand on the other side. All right, that's the complement. And then it's the same here. You're making a complement, but whenever you put a T, you're going to put a U, and it's not going to stay attached. Of course, the RNA is going to float off and be a single-stranded molecule. So over here, you're making a double-stranded molecule, double-stranded DNA. But here, you're kind of unzipping you may think of it this way, unzipping the DNA, and you're just going to make a copy of this here, and that copy isn't going to stick. It isn't going to be like this up here. It's instead going to be able to float away as RNA. All right, so this is your RNA here. This is DNA. And of course, this is double-stranded DNA. This is double-stranded back here. It's single-stranded up here. All right, and of course, we also need to know that what is the molecule that does this. That is Rna Polymerase. And we'll talk about 1, 2, and 3. There are 3 different versions of it. But Rna Polymerase, I'll just abbreviate it POL. That's fine. I'll always accept Rna Pol. It's abbreviated in the books, it's abbreviated in research papers, etc. So it's okay to abbreviate it RNA Pol. And RNA Pol, of course, is a protein that will attach to the DNA and make a RNA copy. A transcribed copy is the way we would say that. Now, later on down here, it talks about TATA boxes. Oh, that's the other page. And realize the TATA box always comes before the gene, and it is where RNA polymerase attaches. This is called a promoter, and we will talk a lot about this in the in about the third week, maybe even next week. We'll talk more about these, but the TATA box is a place where RNA polymerase can attach, and TATA is one example of a what we call a core promoter. It's a sequence that will attract RNA polymerase, and of course that means that's where a gene is, and that all genes begin with TATA boxes. All right, so there's some information for you at moving forward. It's in the videos, but it's kind of subtle here in the first couple of weeks. You may not have quite caught on when I mentioned it in the other videos. Compare and contrast number 1 and number 2. There are many answers to this. I'll accept almost anything, but what are the differences? Here we're copying the information to make sure it's preserved. Here we're copying the information so that it's used. So that's the main idea here. Yeah, you can write other things and they're correct. If anything you write here is correct, I'll count it. But really the idea here is when you copy DNA, you're copying DNA to preserve the information for usually the passing it on to another generation, to a new cell. But usually when you're making an RNA, you're copying the information in order to make something for that cell, within that cell, for it to function. All right, because RNAs, it's not the only function of an RNA, but the primary function of RNAs is to send it to a ribosome so the ribosome can read the instructions and make a protein. So realize genes are instructions for making proteins. Actually, I shouldn't say that's not their only definition of a gene, but that was the long-standing one, something we called the Central Dogma. And we now know that most of the things in the Central Dogma, there are a million exceptions to. So Central Dogma is kind of out of fashion. But generally it was thought one gene, one protein. And we already know that with splicing, if you know what splicing is, one gene does not necessarily mean one protein. It means, well, one type of protein maybe, or one part of a protein. So there are lots of things wrong with the Central Dogma, but it's what we went by for like 50 years. So you have to learn kind of the basics here. And the idea here is that one of these is for ensuring the integrity of the genome going forward. So you're going to copy it double-stranded, make sure it's safe, pass it on to the next generation. But down here is when you're using it. All right, you're using it to has to do with its function. Up here, this one has to do with the preservation of the knowledge. All right, now here's where I saw many of you making mistakes in a couple of the worksheets you turned in. It says, what is the position? Well, the position is the number. So 1, 2, 3, 4, 5, 6, 7. So I'm looking for the position of the 3 nucleotides that make up a start codon. So this is trying to figure out if you know what a start codon is. If you don't know what a start codon is, it's an A, U, G. So I want to know if you know what a start codon is. And of course, is there an AUG? Well, you don't know from looking at these. You have to make the complement first. So in other words, if your answer here has an AUG in it, then you would know that you have a start codon. So what would code for an AUG in your answer on number 2? Well, that's going to be a T, A, and a C, and I'll leave it to you to see if you have a TAC in there. And if you do, then you tell me the number the three numbers that make up a TAC. So that's what I was getting at here, and that's just a test to see if you, so the numbers down here are the positions, and so I'm asking you for the three positions of a TAC. There is not a stop codon, so this is also a test to see if you know what start and stop codons are. If you learned that, you should have learned that in high school, but maybe not. What is the position of the stop codon if there is one? So you need to, it actually says there isn't one, so it gives you that hint. So nucleotide change, in other words, a mutation, how would you get one? So that's kind of a, it's not an impossible thing to figure out, but you just look up here and figure out if you can get a stop codon up there. Now, keep in mind that a stop codon have to be in what's called the reading frame. So make sure you watch, I'm not going to be able to read, reading frame. I think you get the idea. Make sure you watch the videos or look up somewhere, find out what the reading frame is. And so if you have a start codon, I will give you a hint. The start codon is going to set the reading frame, which is going to help you figure out number 5. All right, so let's find the other half of the worksheet here. Oh, this should have the second page. How do I get to the second page on this program? Oh, this is a brand new program to me, so do not know. Nope, can't go that way. Down here. Nope, I'll have to reopen maybe. Nope. I was sure there was another page. Maybe, oh, here we go. That program just wasn't obvious where to go. Now on this one, I'm going to lose the drawing ability though. On this one, translate, so you need to know how to use the genetic code. Translate means turn it into an amino acid sequence. And that means you read these letters up here 3 at a time, starting, this is the reading frame, starting with maybe your start codon. I just gave that one away. And then you read them 3, 3, 3. That's a reading frame. And what I want you to know is translate the mRNA sequence with the stop codon. In other words, once you changed it and you translated it so that it had a stop codon, give me the amino acid sequence that resulted. Well, I'm giving you a hint here. It's going to be 2. There are going to be 2 amino acids created. If you don't have a stop codon, without a stop codon, it just keeps reading until it runs into one. So that means you're going to read the whole thing after, starting with the start, you're going to read the whole thing. And I'm giving you the hint here that that's going to be 1, 2, 3, 4, 5, 6, 7. So you're expected to have 7 amino acids here. So here you're giving me amino acids, and you can use that chart. You can use single letters or you can use 3 letters. I like 3 letters, but you know, for writing this one, single letters are fine. So just realize these are numbers. These are going to be amino acids. All right. Now there's a mutation that's going to insert a C between positions 3 and 4. So here is 3. So it'd be like putting a C right between these two. So whatever you have here, you're going to add a C here. So if you go up here and copy your sequence, starting with number 1, and you basically, those are the same, yes. So it'd be like 1, 2, 3, we're going to put a C right there. All right, so that's what you're doing here. Now you reevaluate and tell me what happens. That's what I want to know. All right, and then what is the translated product if that occurs? It's to show you that some mutations are more important than others. Some mutations, any mutation that changes the reading frame is going to make a completely different program, a completely different protein. So that's what I'm trying to emphasize here. and then it tells tell me if anything except for a C had gone in there. So that goes back to knowing kind of what a start codon is. So that's a little hint. Now, and then it says using the original sequence in number 1, add a TATA and a termination sequence at the appropriate places. Also indicate the 3' and 5' orientation. Now, one way to answer number 9 is actually to read worksheet 2. If you read worksheet 2, it actually will tell you the answers to this one. But a TATA is a promoter, and it goes at the beginning of every gene. Now, the location can change, so I'm not going to ask you where the TATA is at the beginning. It just comes before the start codon. So as long as you put a TATA before the start codon, then you get this one right. Now, as what is termination sequence? Well, I'll tell you what. Go to worksheet 2 and read worksheet 2, and that will give you the everything you need to know. There are also videos on this, but I'm not trying to make this a hard worksheet. I just want to see what you already know, and I want then, if you don't know these things, I want you to be familiar with the implications of changing the sequence. In other words, in this one, inserting a C right here is actually a bigger deal than inserting a C down here, or I should say, actually it's not. But doing it, inserting something other than a C is very important and actually will mess things up a lot. So not only the location, but the exact letter that you insert, or you mutate, you alter, delete, change the letter, however, whatever you want to call it, location and exactly what happens, whether or not you delete it or change it to a different letter or add one. All of those have different implications, and those are the things you need to learn first in this class. What are the implications of changing this particular position? Why is this particular position important? Well, you should get that out of this worksheet. And then, of course, adding one in the middle, what does that do? Well, that changes the reading frame. Changing the reading frame, of course, is going to alter the protein quite a bit. Anything after the alteration is going to be different. So make sure you're aware of those things. Those are the things I want you to get out of this and the next worksheet.